Hello students, I am Assistant Professor Aparna Kedari and today we are going to study dyes and pigments. So first definition of dyes. A dye is a colored substance that has an affinity the substrate to which it is being applied. Then dyes are substances capable of coloring fabrics in such a manner that the color cannot be removed by rubbing or washing. It should be noted that every colored substance is not dye. Then, for example, isobenzene. Isobenzene is of orange red color, but it is not a dye because it is not capable of coloring the fiber. Dyes can be used for imparting color to the paper, plastic, and leather. Then there are some qualities of good dyes. The qualities of good dye, it must have a suitable and attractive color. It must be able to attach itself to materials, that is, for example, cloth, from solution or to be capable of fixed on it. For example, isobenzene is a colored but cannot fix itself to a fabric. Therefore, isobenzene is not a dye. Picric acid is able to dye silk or wool a permanent yellow color but not cotton. Hence, dye must form a chemical union with substrate to be dyed, or it may get associated with the substrate by an intimate physical union. Dye must be soluble in water or in suitable solvent and should form a stable or good dispersion in water. Next quality of dye, good dye is the substrate to be dyed must have natural affinity for appropriate dye and must be able to absorb it from solution or aqueous dispersion. When a dye is fixed to substrate, it must be fast to washing, dry, cleaning, light, heat and other agencies. It must be resistant to chemicals like soap, washing soda, detergent, acids, and alkalis. Color constraints chromophore exochrome. According to Grave and Lieberman in 1868, unsaturation is essential for light absorption. They pointed out that color was due to unsaturation and conjugation of alternative bond and double and single bonds deepens the color. Saturated compounds usually appear colorless because of fact that they absorb ultraviolet and infrared light. Unsaturated compounds containing carbon oxygen double bond, carbon nitrogen double bond, carbon carbon double bond, carbon sulfur double bond, nitrogen nitrogen double bond, nitrogen oxygen double bond, carbon nitrogen double bond, particularly in conjugation, absorb in visible region of the spectrum. So as a result of which they are expected to appear colored. Then, according to O.N. Witt in 1876, pointed out that all colored organic compounds contain certain unsaturated groups which are responsible for color, and he called this group as a chromophores, that is chroma color. Phoras means bearing, that is color bearing substances or compounds. Thus, group responsible for selective absorption of light of wavelength 2500 to 10,000 angstrom are called chromophores. Now, NO2 group, then that is nitric oxide group, nitrogen dioxide, nitrogen double bond, carbon carbon double bond, carbon oxygen double bond, and carbon or sulfur double bond, etc. A compound containing a chromophore is called chromogen. If a chromogen has one or more exochrome, the resulting substance is called dye. Therefore, dye is the combination of chromogen plus exochrome. Then here there are types of chromophores. First is the independent chromophore. When a single chromophore is sufficient to impact color to the compound, for example, azo group N double bond N, nitroso group nitrogen oxygen double bond, ortho and para quinoid group, quinonoid group, Dependent second type is the dependent chromophore. More than one chromophore is required to produce color in the chromogen. For example, carbon oxygen double bond, carbon carbon double bond group, acetone having one ketonic group and is colorless, whereas diacetyl having two ketonic group is yellow in color. Triketopentane having three ketonic groups, therefore it is orange in color. Here you can see this colorless acetone, and then yellow diacetyl, that is because it contains two carbon oxygen double bonds in conjugation. Here three carbon oxygen double bonds in conjugation. So, what makes the dye? Next, moving to what makes dye colored. Dyes are basically ionizing and aromatic compounds. They have chromophores, isolated functional groups capable of absorbing UV radiations. Example: nitro, N nitrosone, N double bond, N carbon oxygen double bond. Then carbon nitrogen double bond, carbon carbon double bond, carbon sulfur double bond present in them. Their structures have aryl rings that has decolorized electron system. These structures are said to be responsible for absorption of electromagnetic radiation that has varying wavelength based on energy of the electron clouds. Then chromophores acts by making energy changes in delocalized electron cloud of dye. This alteration invariably results in the compound absorbing radiations within visible range of colors and not outside it. Human eye, as we know, human eye detects this absorption and responds to color. So how can color of dyes be altered? We can alter the color of dyes. The color of dyes are altered by the modifiers. The color modifiers of methyl and ethyl groups are responsible for any alternation in the dyes. They alter the energy in the delocalized electron. For example, methyl violet series. We can alter the color here. First, there are six methyl groups. Here, reddish purple color. This compound is purple blue. Here, methyl green color. Therefore, we modify the color of the compounds. Then exochromes. Exochrome is the only substance that responsible. Exochrome, the only substance responsible for providing solubility and cohesiveness to dyes. And exochrome is a group of atoms attached to chromophore which modifies the ability that chromophore to absorb light to a longer wavelength. We, uh, we call these groups as exochrome. That is Greek exon means to increase and chroma means color. Some of the exochromes are OH, OCH3, NH2. That is here, this benzene ring is a chromophore, shows wavelength absorb at wavelength 255 nanometer. When OH group is attached, that is phenol, absorb at 270 nanometer. Then anisole 269 nanometer. That is OCH group is a exochrome.